Anyone want to take a stab at it? So what does this show?
Well, it's showing that as we continue to shake everything up in the blender.
Blender?
I think of it as a blender.
I think it is a blender.
Blendor.
I didn't even see that.
Wow, I didn't even notice that. That's funny because this is actually swiped from like a textbook. That's pretty hilarious.
But there's much less of phosphorus 32 that becomes extracellular relative to the sulfur 35 and the infected bacteria.
Mm-hmm.
So because of that, we can make a conjecture that there's I lost my train of thought. But it's the phosphorus 32 radioactive, causing radioactive DNA that's actually affecting the mice or whoever the person.
Well, in this case, we're just using bacteria and viruses.
Okay,
right?
Well then the bacteria,
Uh-huh.
because then there's more of that intracellular
Yeah.
than it is extracellular.
Yeah. So just to clarify one thing, we're in this experiment. You're definitely right on that. But in this experiment, we're not interested in the effect of radioactivity at all. We're simply using the radioactivity as a marker to trace the location of protein or DNA. So it's basically just like a tag you can detect with a Geiger counter or a similar type of approach to see where it went. But yeah, so you can see that when you start the experiment, you have, essentially you have maximal infection. So infection happened, injection of information occurred. Then you shake them using the blender, and a small amount of the phosphorus becomes extracellular, but a solid 70% of all the DNA that had the radioactivity that went into the experiment remains inside of the bacterial cell, which is amazing. I mean, that's almost all of the radioactivity is still inside the cell. As you shake off the bacteria, as you shake off the bacteriophages, you shake off more and more of the protein-associated sulfur, but it doesn't affect the infection because the infection is based on the phosphorus, which is associated with the DNA. So I know that last step may have been a little bit confusing. The key thing to notice here is almost all phosphorus is intracellular. Almost no sulfur is intracellular. And so this experiment was pretty much like the nail in the coffin of the idea that it was protein and not DNA that carried the heritable information. And after this, you were kind of a crackpot to sort of think that it was protein and not DNA. Now there's a big leap here, right? Which is, what are we studying?
Bacteria. Right?
We're studying viruses of bacteria to understand heredity, right? So the big leap here, of course, is what?
Does it work with people?
Yeah, is it the same thing in humans and mice and plants and bacteria? I mean, is it the same everywhere? Or is this just a particular little piece of the puzzle where it works this way? And there's reason to think that it would be, given that we have viruses also, that are also just protein and DNA that infect us. And they probably inject information of a similar type. And so, but that is sort of a connection that isn't necessarily true. Of course, it's turned out to be accurate, but that was an extension of this that wasn't necessarily known at the time. Okay, so once they figured out that DNA was it, that it was the molecule that controls pretty much all of heredity, the race was on to solve the structure of DNA. And so I want you guys to get out the worksheets that you had, and if you guys can maybe assemble yourselves into small groups and discuss those questions from the Secrets of Photo 51 video, I will check you off as you discuss.